holding it until access.
We are right at noon, everyone. If I could have everyone's attention, please. I want to welcome everyone to a new semester of Grand Rounds. Really great to see everyone out. We have a really special treat today. We have a speaker from New Zealand, Dr. Horsfield, and as it kind of worked out, Dr. Merriman, who works in the Department of Medicine here, helped arrange us to have this speaker today. And he knows her really well. He's actually going to do the introduction, so I'm going to step aside here in a second, and then he's going to hand things off to Dr. Horsfield here in a couple minutes.
Thank you very much, and thank you to the Division of Pathology for giving Dr. Horsfield an opportunity to present. So I'm Tony Berryman, been in the Division of Rheumatology, Clinical Immunology and Rheumatology for about 3 years now, all the way from New Zealand. But Dr. Horsfield is a long-time friend and colleague of mine, and she has, introduced me into these experimental systems which we use in the context of gout, but she'll be talking about that in the context of cancer today. So she did her undergraduate in Victoria University, which is in Wellington, New Zealand, and then came down to Dunedin to the University of Otego, where we cohabited doing PhDs at the same time. So that's how I got to know Jules. So your undergraduate was in botany, right? And a degree in botany and a PhD in biochemistry in 1995, and then she went to Adelaide for a postdoc, and then after a few years there, we spent 8 years in Auckland, finishing off as a research fellow in Auckland, where she was working with a established or as you've got saying zebrafish, but you're saying zebrafish, so excuse whichever pronunciation you decides to use. And then in 2007, she came down, where she did a PhD from at the University of Otago, where she is now the head of the Department of Pathology. And in New Zealand, the hierarchy between departments and divisions are sort of inverted. So a department is roughly equivalent to a division here. So you hand over to Dr. Horsfield, and thank you so much for coming along and doing this.
Thank you, Dr. Merriman, for that very kind introduction. Well, kia ora koutou. This is how we say greetings in Aotearoa, New Zealand, where I'm from. I'm absolutely delighted to be here in this lovely city of yours, Birmingham, and I had to be, I had to learn how to say this in American, and and it's a thrill to be able to talk to you today about some of our research. And I won't be talking to you today much about zebrafish research. So a lot of what my lab does is developmental biology in zebrafish. But today I want to tell you some stories about how we're looking at how you can drug chromatin mutations in cancers. And most of the work comes from cancer cell lines and sort of drug screens using those types of models and cell lines with a tiny little zebrafish vignette at the end. So this is called Drugging the 3D Genome, Strategies to Combat Cancers Driven by Cohesin Deficiency. So if we haven't heard of Cohesin yet, we soon will in this talk. Here's a picture of Queenstown, New Zealand.
So we get the bar on. Is it
This
okay?
one?
Yeah.
I have that.
There we go.
Yeah, okay. This is a lovely picture of Lake Wakatipu in Queenstown, and it's just a nice advertisement for anyone who would like to come and visit at the University of Otago, which is about a 3.5-hour drive from this beautiful lake. We would love to host anybody who wants to come and visit the Department of Pathology in Dunedin, New Zealand. Right, so we're going to zoom right into the nucleus to start this talk, and many of you will be familiar with the packaging of DNA, and so first we need to wind our DNA around the core histones that forms the beads on a string chromatin fiber. But then there are higher levels of organization of DNA still. So for example, I need to click the mouse. So we need to make these chromatin loops, and then we can organize the loops further into sort of globules of DNA known as topologically associating domains. And these domains then are further organized into chromatin compartments, which form the chromosome. Whoops, I need to go back. How do I go back? This should not work. Oh yeah, I got it. Okay, so you'll notice that the chromosomes in the nucleus also like to hang out together. So chromosomes occupy chromosome territories in the nucleus. So really you've got all these different levels of three-dimensional genome organization in the nucleus. And you might think, well, okay, this is pretty cool. We've got to get 2 meters of DNA into our nucleus somehow, and we've got to package it somehow. But this packaging turns out to be really quite important for how genes are regulated in the nucleus. So many of you might be familiar with euchromatin and heterochromatin compartments in the nucleus, where you have areas of activated genes and repressed genes. And so how we organize this chromatin turns out to be really important for normal cell homeostasis, and it can go horribly wrong in diseases such as cancer when we get a lot of chromosome rearrangements and scrambling of the nucleus. So just to kind of zoom in a bit more of the technical and organizational principles of these chromatin looping events, just wanted to focus in on these topologically associating domains to start with. So these are domains of contact in the nucleus where pretty much DNA sequence inside these topological domains can contact everything else inside that domain, but not outside. So if you think of it like two high schools, a girls' high school and a boys' high school next to each other, you've got a lot of interactions amongst the students in the high schools, and you've got a big fence down the middle to make sure the boys and the girls don't interact inappropriately. So in Dunedin, we have Kings and Queens high schools, and that's the situation there. I don't know if you have the same situation in the USA, but think of a topological domain like that. You get lots of enhancer gene interactions inside the domain, but really the domains are designed to keep things apart that shouldn't be interacting. And the proteins that are involved in this are known as Cohesin, the Cohesin complex, and the CTCF insulator factor. So CTCF is a sequence-specific binding factor that will bind to CCCTC binding sites. And Cohesin is basically anchored at these CTCF sites to make these loops of chromatin that keep interactions inside that domain. So a lot of what people, so how people really found this out is by doing experiments such as high C. So high C is a measurement of what in the nucleus interacts with what else. And the readout of a high C looks something like this. So this is a heat map where the bright red pixels that you can see correspond to regions of high interaction between different sites. So if you look at this pixel here, this represents an interaction between this site on a linear chromosome with this site over here on a linear chromosome. So we take these readouts and interpret them as which piece is interacting with what other piece. And these topological domains, as I said, are organized by Cohesin and CTCF. So Cohesin's got some other jobs as well. It got its name Cohesin because it was first discovered for its role in holding together sister chromatids during mitosis. So in the Cohesin cycle, you have this large ring-shaped complex, which is Cohesin. It has 3 basic core subunits, structural maintenance of chromosomes complexes 1 and 3, another subunit called RAD21, and then an associated stromalin or STAG subunit. And these can be modified by events like acetylation and phosphorylation. And in normal interphase cells, the cohesin complex is cycled on and off of DNA by other factors. And so loaders for cohesin are this complex NIPBL and MAL2, and then unloaders are PDS5 and WAPL. So if you just think of it as complexes cycling on and off DNA, and when the complex lands on DNA, it performs a task known as loop extrusion. So if I sort of demonstrate with my scarf what loop extrusion is, if this is your cohesin complex, then the DNA will be extruded through the complex like this until it hits a CTCF site here. And that's how you get your loops of DNA. So in every one of your cells, all of the time, there are zillions of cohesin complexes busily extruding away the DNA until they hit CTCF sites. And so you can get loops upon loops upon loops, and this is happening constantly. And the complexes are unloaded and loaded constantly as well. So when we take the high C experiments, we're taking an average of all of these loops that are formed by cohesin complexes, but really it's a very dynamic process. So this is happening in interphase. But once you come to S phase, when DNA starts to replicate, cohesin becomes stabilized on DNA, and it's stabilized by acetylation of, and the enzyme that does that is known as ESCO1 or 2. When this complex is stabilized, it stays on the chromosomes, and now it holds together the sister chromatids of the replicating cell. And it can still be removed, which it is sort of later on in pro-metaphase by some other proteins here. But eventually, when you get to mitosis, an enzyme called separase cleaves the RAD21 subunit that opens the cohesin ring, and that releases the chromosomes to move to opposite ends of the cell in mitosis. So, so that's a very important role that cohesin has in cell division. Some of the subunits are recycled in the next cell cycle, and then some, like RAD21, have to be resynthesized. Let's not do that. Let's do this instead. So you've got two very important consequences and functions of cohesin in a cell. So the loop extrusion function obviously contributes to chromatin organization and hence gene expression. But the sister chromatid cohesion function contributes to progression through cell division. So one important question I think that's out there is, can these two functions really be picked apart? Particularly in cancer, when you've got both of these things going on, cells obviously have to divide, and then they've got a, particular oncogenes need to be dysregulated for cancer to happen. So, so how are these linked, and are they linked? And so these are some questions that we're trying to answer. So because this ring does all these wonderful things, this is how I like to see it. It's the ring of power. And it controls these very important processes. So earlier on in the cohesin, in research on cohesin, people used to think that some of these looping events were really hardwired. So this is sort of before the principles of loop extrusion were known. People thought that perhaps cohesin binds with CTCF and makes these solid, unperturbable loops that kind of sit there and insulate domains. And perhaps it actually hardwires enhancers to promoters of genes and forces genes to be expressed. And with RNA polymerase and say the mediator complex. And so some of the work that we've done has really been part of research that's challenged this view. So aside from cancer, if you have germline mutations in cohesin that are haploinsufficient, not null, because if the mutations were null, they would be non-survivable. If you have haploinsufficient mutations in cohesin or its regulators, you can wind up with a spectrum of developmental disorders known as the cohesinopathies. And these include disorders like Cornelia de Long syndrome and Roberts syndrome. And I'm aware that there's a really good organisation for Cornelia de Long syndrome in the USA. And I've come to some of the meetings for that, and it's been really amazing. I'm only aware of one patient in New Zealand with Cornelia de Long syndrome, because we just don't have that many people. So it's really good to come here and interact with the communities here. So, so understanding the basis of this syndrome is another important part of what my lab does that I'm not going to really talk about today. So these are caused by germline insufficiency mutations. But in cancers, cohesin mutations pop up in all kinds of different cancers, particularly STAG2 mutations. So STAG2 is one of the important regulatory subunits of cohesin. So STAG and cohesin mutations are in a variety of cancers. And then these somatic mutations that pop up during the evolution of tumours. So how I first got started in this area of research is when I was working at the University of Auckland. I was recruited to work in a leukemia lab, and this lab was interested in understanding how blood development gets subverted to produce leukemia. And an arm of research in the lab is really interested in how cells divide, commit to their fates, and undergo terminal differentiation. And a key gene that is responsible for this terminal differentiation is RUNX1. So along the myeloid and lymphoid lineages, RUNX1 is activated at particular times, and also in hemangioblasts to HSCs. So as many of you will be aware, mutations or translocations that involve RUNX1 cause leukemia, particularly acute myeloid leukemia. But these mutations are not on their own sufficient to cause leukemia. And so when I was hired by this lab, what we were trying to do is to look for second site mutations that would cooperate with RUNX1 to give you leukemia. And we used a zebrafish model for this work. And how many people are familiar with zebrafish as a model? Some of you? Excellent, great. So, so they're really handy because they've got very visible embryos. And so for anything imaging to do with development of organs and systems, you can look at extremely easily in a zebrafish embryo. So what I'm showing you here is very early zebrafish embryos, just when germ layers are sorting themselves out into cell populations that give rise to blood and neurons and muscles and such. And here in this embryo, the dorsal view of this embryo, you can see that there's a band of cells on either side of the embryo that expresses RUNX1, and these will later become hematopoietic progenitors. So this is the dorsal view. We're looking down on the back of this embryo at the lateral plate mesoderm here. This is the lateral view, and you can see RUNX1 expression in the lateral plate mesoderm, and also this anterior lateral mesoderm here. This expression, that's punctate, that's more towards the center of the embryo, that's actually neuronal expression, and this is the hematopoietic specific expression. And in the mouse, it's sort of equivalent to the AGM expression of RUNX1, which marks the early fetal multipotent blood progenitors. So I performed a forward genetic screen using a RUNX1 in situ hybridization as my readout. And I was looking for second site mutation that would then affect the expression of RUNX1 in hematopoietic tissues. And I did find a mutation that did just that. So here you can see a normal embryo. Here's the expression in the lateral plate mesoderm that will become blood. And you can see that it's surrounded by embryos that don't have this expression. And I called this mutation 17.1 because it was the first fish from the 17th batch of fish I had screened that, and it was, yeah, so that's why it's called that. And then, and this is a ways back, so this is kind of 2002 or so that I found this. It took another 2 years to find out what this gene was. Some of the clues to what this gene was, though, came from my previous work in Drosophila, where I was doing forward genetics on cell cycle mutants. And it prompted me to look at, well, what might be wrong with these embryos, as well as having RUNX1 that was disturbed. What I could see in the embryos once they got a bit older is that the chromosomes were condensed and all over the nucleus. And it looked very much like the cells were being stuck in the metaphase-anaphase transition. And when the mapping regions of zebrafish finally got good enough to look to see what genes might be good candidates, it turned out that this gene called RAD21 popped up in the region that I was mapping. And RAD21 is a core subunit in mitotic cohesin. So what I was looking at in these embryos was cell cycle disruption. So maybe it was this gene. And it actually, once I kind of cloned and sequenced it, it turned out to have a nonsense mutation that was causing this mutation. So here you can see early in development, there's the mutant phenotype of RUNX1 non-expression in hematopoietic tissues. But later you can see that these embryos die, and they die because they can no longer divide their cells anymore. There's two things going on. A really specific problem with gene expression, because the neuronal expression is preserved, and then a big problem with cell proliferation later. So you might be asking, well, why are you seeing, how come they're lasting that long? And it's because if you notice this big kind of globby thing that the embryo is sitting on, that's the yolk of the zebrafish. And the yolk contains a lot of mRNA that's maternally deposited. And that maternally deposited mRNA sees that embryo through several cell divisions before it finally runs out of RAD21 and stops in the cell cycle. But the time at which it stops expressing RUNX1 in blood cells and blood progenitors is before that RAD21 runs out. So that means that the dose of RAD21, therefore cohesin, is crucial to maintain the expression of that gene in blood cells. So this was the first real link of the cohesin complex with a gene that's involved in leukemia, well before genome sequencing projects identified cohesin mutations in leukemias, and then a whole bunch of other cancers as well. And because the RUNX1 expression disappeared at a time when cell division could still occur, it was the first evidence that dosage of cohesin is really important for maintaining expression of this gene. It was really hard to publish this work, actually, because people were like, well, what's the mechanism of this? And got to remember, this is like nearly 20 years ago now. So nobody done any high Cs, nobody knew what cohesin did other than cell division. So really, this is when I started my lab, and since then we've been trying to understand what cohesin actually does in cancer and development. And if we were done yet, I'd be out of a job. So yeah, there's a lot to discover. So what's the pathological mechanism of these cohesinopathies? So at the same time that I published this work, it was found that mutations in the loader for cohesin, NIPBL, was responsible for the major gene responsible for Cornelia de Long syndrome, and an acetylase for cohesin was responsible for Roberts syndrome. And so really the whole field took off as to what these things are doing in development. And one of the first things that we did in my lab is to have a look, well, is the role of cohesin really just restricted to proliferating cells? And the answer to that is really no, because when we looked in tissues such as brain, we could see that cohesin expression is well outside of the zones of proliferating cells. So here's a section through a zebrafish brain. Here are the proliferating cells as marked by pCNA. And here's where RAD21 is expressed. So and those are all post-mitotic neurons, right? So if it's only needed for proliferation, what's it doing in these post-mitotic neurons? So we surmised it might be in the regulation of gene expression back in 2009. And then when we took our zebrafish embryos and performed microarrays, because that's what you did before there was RNA-seq, we found that there are many gene networks dysregulated in RAD21 mutants. And amongst them is the oncogene MYC. So that's actually down-regulated along with a whole network of other genes related to MYC in cohesin mutants. And however, what we could see is if we made knockdowns of some of the other components of the cohesin apparatus, such as ESCO2, which is the Roberts syndrome gene, we find that we don't get the same gene regulation patterns at all. So this was the first indication that not all of the components of the cohesin apparatus function in the same way in gene regulation. And if you remember ESCO2, it's the acetylase for cohesin that stabilizes it in mitosis. And we later went on to show that the Roberts syndrome is really different from Cornelia-de Long syndrome because it's got much more of a cell cycle phenotype as opposed to a gene expression phenotype. So back to cancer. More recently, we've been looking at what genes and signaling pathways are affected by cohesin mutations in cancers. And we started out, because of identifying the MYC gene in the microarrays, we started out wondering, well, is cohesin essential for MYC regulation? And maybe you could get overexpression of cohesin in cancers, and you do in some cancers get overexpression of cohesin. And could this be a pathway to oncogenicity in particular cancers? And then we were operating on the principle that maybe the enhancer promoter looping that you get to regulate genes is a hardwired thing. And you need cohesin to make these loops between, say, enhancers for MYC and the MYC gene. And then this paper came out in 2010 that actually identified that cohesin binds through the genome with the estrogen receptor at particular genes that are estrogen responsive. And this paper was really the first to show that cohesin bound elsewhere other than with CTCF in the genome. And so it could bind with transcription factors and have a transcription regulation function. And so their hypothesis was that estrogen receptor and cohesin form these loops that linked genes with enhancers. But if you look at some of their data, what they do show is that genes bound by estrogen receptor and cohesin and not with CTCF are much more sensitive to estrogen stimulation. But if you look here, there are more genes that are down-regulated than up-regulated by this. And that's kind of intriguing, isn't it? Because if your idea is that you're linking enhancers and promoters, why are you getting down-regulation of particular genes? So we thought we'd test this because this paper for some reason did not do this. And then we found out why they didn't do it, and that's because it's very confusing when you do. And that's to take out cohesin and then stimulate with estrogen and then see if you lose all of the estrogen responsive genes. Like, that's the obvious experiment, right? I was like, well, why wasn't this in this paper? Let's do the experiment. And oh my God, it was so confusing. So a lot of genes didn't care whether you took out. A lot of estrogen responsive genes didn't care. Some of them went up, many of them went down, and yeah. So anything could happen, really. But when we looked at MYC, because we were really interested to see, well, is this MYC is upregulated by cohesin and estrogen? So do we end up getting a linkage that upregulates MYC? So we took MCF7 cells, we took out cohesin, and we stimulated with estrogen. And this sort of clumsy diagram shows you where estrogen binds in pink, estrogen receptor binds in pink, where RAD21 binds in response to estrogen in blue, and where RAD21 binds anyway. And then we did an experiment known as 3C. So this isn't the fancy high C experiment where you're doing everything with everything. This is testing fragment by fragment which bits will link with the MYC gene. So there's an enhancer for MYC about 66 kb away from the promoter. And we found that if we tested that interaction when you knock down RAD21, you decrease that interaction. So this cartoon is just summarizing a lot of qPCRs that aren't very interesting to show you. So yeah. So we're like, yay, this works. So if you take out cohesin and stimulate with estrogen and you lose the loops that drive MYC expression, that's exactly as what you predict. But Again, many of the genes are downregulated and this is an essential thing. So do all genes require cohesin to link enhanced promoters in the same way? And what made this particular work really hard to publish is that some loops got better without cohesin. So here's another chromosome. There's a chromosome 21 locus, the trefoil factor locus. Some of you might be familiar with this locus in cancer. So there's a kind of an array of TFF genes, and then this one, TMPRSS3. And what this really shows is that this is cells that are in cohesin intact, and this is cells that are cohesin deficient. When you knock down cohesin, the expression of some of these genes goes up in response to estrogen, which is not what you'd expect, right? And this is like 3 and 6 hours following estrogen stimulation, showing that this is reasonably direct. So it's not like it's a secondary effect of something else switching on these genes. And this is where these genes are located along the chromosome. So if you do the similar experiment, just let me go back a second. We could show that for this gene here that goes way up, there's enhancers for it that lie within the trefoil factor 1 gene. And the loops actually get stronger and better when you take out cohesin. So what's going on? I mean, this should be impossible, right? Because you need it for the loops. So imagine trying to submit that in a time when you're supposedly having to show that this is a complex that makes loops. So the highlights, we did eventually get it published, and the highlights are that genes can do anything they like when you take out cohesin and stimulate the estrogen. You can enhance a promoter communication when you take out cohesin and stimulate with estrogen. You don't need it to anchor ER estrogen loops at all. And sometimes you might need it to restrict the formation of these loops from happening. This made much more sense much later when loop extrusion was discovered as the principle for genome organization, and that the insulatory principles of these topological domains was discovered. So if you can imagine that loops insulate things, you know, the high schools from interacting, you take out some of that insulation, you're going to get some interactions that are inappropriate. So I think that's what we were seeing in this experiment. But at the time, it was tricky because it was assumed that the looping was hardwired. But it's not, and then we see deconstraint of transcription. So this is interesting for cancer though, because you can imagine a scenario where you've got to be a normal cell in normal conditions. You've got to have everything under reasonable control, and so if you release the brakes on some of this control, you're going to get some of these events that can stimulate cells to take off. So now over to leukemia, which is also on chromosome 21. We started to think about following up on the RUNX1 finding and modeling leukemia. And so we developed some standard cell line models for this. So out of zebrafish now and MCF7 cells. So we chose cell lines, erythroleukemia cell lines, and then the CMK cell line, which is a Down syndrome megakaryoblast. And the interesting thing about that cell line is it has 3 copies of chromosome 21. So that makes it interesting for us to study, considering we're seeing all these different events of looping on chromosome 21. And we also used another cell line known as MCF10A. So that's a breast epithelial line that's immortalized, but it's not a cancer cell line. It's just been, I forget what's been done to it, but it's been given a kind of thing to keep it going, but it doesn't have any cancer mutations. And so what we wanted to do was to use these cell lines to look at the mechanism for gene expression changes. And this one, we made deletions and then used that to find synthetic lethal targets, synthetic lethal drugs for Cohesin. So just to talk about the mechanism for a little bit. One of the mutations that's most common in leukemias is STAG2. And postdoc Jesha Anthony in my lab made a STAG2 mutation that phenocopies STAG2 mutations that are commonly found in leukemias, so an amino acid change that introduces a stop codon. And then she did an experiment known as a TAC-seq. So this experiment basically assays for open versus closed chromatin in the genome. And if you see differences in accessibility, then that tells you something about what regulatory factors can come in and bind and regulate gene expression. And what she found is that there's many differentially opened and closed sites when you make this mutation at baseline in the cells. And a lot of these sites are in locations known as super enhancers in the genome. So super enhancers are kind of large tracts of DNA that attract a bunch of transcription factors and are thought to be master controllers for many different genes. So differentially increased attack sites were at super enhancers, meaning that these cells are much more open for business when it comes to activate particular genes. So let's have a look at RUNX1, which kind of started the whole thing. We could find, so here's the RUNX1 gene. It has two promoters, P1 and P2. There's a large intron between it that contains super enhancers, and these super enhancers become more open in Cohesin mutant leukemia cells. And this just shows you where the differentially opened attack sites are down here and how they coincide with super enhancers here. So what are the consequences of this? So when we did RNA-seq on these K562 cells, there wasn't really like a massive difference between Cohesin STAG2 mutant and wild type cells. So Jesha had the idea to, well, what happens if you make these cells do something? Maybe if they're just sitting there dividing, they don't really care about STAG2 mutation, but they might care if you stimulate them to do something interesting like differentiate. So she put PMA on the cells, which differentiates them down the megakaryocyte lineage. Normally when that happens, you activate RUNX1 expression, but you activate it from the P1 promoter. So that's the far away promoter of RUNX1. And you can see over here in wild type cells, RUNX1 turns on over the time course of PMA stimulation. However, in the two STAG2 mutant clones that she had, RUNX1 did not turn on at all. So it fails to be induced by PMA stimulation. But interestingly, expression from RUNX1 P2 is precociously induced when you have a STAG2 mutation. So this is the promoter that's right next to the gene. And so what this is basically telling you is that there must be some 3D structure that depends on Cohesin to organize this gene to get the right promoter to be used. If you can't use P1, you'll precociously use P2 and have this sort of weird spike in expression, and then a failure of this gene to properly be maintained. So the cells themselves, they don't actually properly differentiate, and they have much more of a kind of an immature phenotype than properly differentiating K562s. So switching to the CMK cells now, Jesha decided to do a high C experiment. So this is the everything with everything experiment now. If you remember in the introduction of the talk, I showed you a cartoon heat map where you can see the pixels. This is what it actually looks like. Hopelessly kind of complex and confusing. And you really have to kind of zoom in on particular regions to see any differences. So chromosome 21, again, here's RUNX1 gene and the ERG gene, which is also quite affected in STAG2 mutants at baseline. You won't really be able to appreciate dramatic changes from this distance. So I'm going to show you a close-up of where RUNX1 is. And even then, it kind of looks very similar. But so these are the topological domains here. And here's the domains where RUNX1 is. And in the wild type, you can see that there are quite defined domains in this region, but in the mutant, you can see that two of the domains are now combined. So you've lost insulation between two of the high schools here, and then you've gained a lot of inappropriate interactions up here, where regions on chromosome 21 are now interacting much more strongly with regions where RUNX1 is over here. And these points of pixels here are not present over here. So that's showing you that there's some inappropriate interactions on this chromosome as a result of the STAG2 mutation, just at baseline in these cells. So remembering too, these cells have got 3 copies of chromosome 21. So one of our hypotheses is that if you have too much DNA and not enough packaging, looping things around to package it properly with, then you're going to get some problems with properly controlling gene expression. However, genome-wide, we see a lot of these differential interactions, and with an experiment like this, it's really hard to know what to focus on because you have so much data. So sticking to this region where RUNX1 is, what we could see is by using another technique called cut and run, which is basically like a ChIP-seq, profiling where proteins bind in the genome. And here is a histone modification, trimethyl K27, and of histone H3. And this is a mark of polycomb silencing. So when you have polycomb activity, you have silencing marks spread through the genome. And you can see that this mark has spread into the intron of P1 and P2 of RUNX1 in these particular cells. And there's another region over here that has more K27 trimethyl than the wild type cells near the ERG gene. And so this change in the chromatin status might account for the change in gene expression that we see when we stimulate cells or get them to do a particular thing. So what we've shown so far is that deficiency and cohesin deconstrained the transcriptional outputs of cell signaling, like when we differentiate cells. At baseline, you can see that the chromatin composition of our STAG2 mutant cells is altered, even without getting them to do something, and that the chromatin interactions, as judged by Hi-C, are also changed at baseline. So basically, you've got a genome that's organized a little bit differently than a wild type genome is. Okay, so knowing all of this means that I guess we know that something changes when we have a cohesin mutation. And again, it has to be combined with other mutations to progress to leukemia or other cancers. And what we really want to know is, can we leverage this sort of mutation to find drugs that will interfere with the biology of cohesin so that we can kill these cells that have cohesin mutations? And so for this, we turn to the MCF10A model. So a Ph.D. student in the lab, Alice Chin, made deletions in 3 of the cohesin genes, STAG2 that I've just been recently talking about, and then 2 core cohesin subunits, RAD21 and SMC3. And she deleted the whole genes for this purpose. So for STAG2, because it's interchangeable with another STAG subunit known as STAG1, you can completely delete STAG2 and make it homozygous, and the cells will still be able to divide. The other subunits, RAD21 and SMC3, the cells would die because they're core components of the cohesin complex. You wouldn't make it through mitosis. So these are heterozygous mutations that were made for this screen. So you've got 2 haploinsufficient cell lines and 1 mutant cell line for this screen. So then we collaborated with some scientists at the John Curtin Medical School in Canberra, Australia, to do this screen, and screened about 3,000 drugs from a company called Compounds Australia. These are FDA-approved drugs. They hit epigenetics regulators, and they're also from PI3 kinase libraries as well. And we basically just assayed cell growth and did the screen as a 5-point dose response screen, and then picked area over curve drugs that gave you a 0.15 differential growth. And then out of that, we had sort of 100 or more candidates that we then subjected to a secondary 11-point dose response screen. And then we followed up with some validation of some of these. So So in the secondary screen, we ended up with about 18 compounds that hit all 3 cohesin mutant cell types. So we really were looking for anything that could compromise all cohesin mutations. And just a reminder of where these subunits are. This is SMC3, a core subunit, RAD21, and STAG2 is the accessory that's interchangeable with STAG1. So here's the list of compounds that were effective. You can see that quite a few of them are cell cycle, like the Aurora complex compounds that inhibit cell cycle. Some are growth factor inhibitors, and many in the PI3 kinase, ACT, and TOR pathways. But there's 2 that we followed up on, because it turns out it's quite hard to follow growth pathway drugs. They're so non-specific, it's really hard to kind of get a handle on whereabouts you can hit things. We've focused on BET inhibition, which is an epigenetics inhibitor, and also this compound here, which is a GSK3 inhibitor, and GSK3 is an inhibitor of the Wnt signaling pathway, so the drug actually agonizes the Wnt signaling pathway. So, interestingly, first to talk about this BET inhibitor. So BET inhibitors inhibit BRD4, which is something that acetylates chromatin and makes enhancers available for regulating genes. And we'd actually already done some work on BET inhibition, but actually not this one particular compound, another one called JQ1, which is, I think, being advanced for clinical use for leukemias already. And JQ1 in combination with PMA actually dampens that precocious spike that you get of RUNX1 expression in the K562 cells. So it reverses the gene expression defects that we saw in cohesin mutant leukemias in our cell line models. It also reduces the stem cell-like phenotype that these leukemias had. Data I haven't shown you is what these cells actually look like in culture. Instead of being nice floaty leukemia cells, some of them sit down on the plastic and then they secrete ECM, and they have kind of these weird stem cell-like properties, including expression of KIT ligand. And if you give these cells JQ1, it reduces the expression of KIT ligand. So you reverse the stem cell properties and you reverse the precocious RUNX1 expression. So now what we can say is that we've got this altered chromatin composition, different sort of epigenetics going on in there, and you can reverse this with BET inhibition, and you can reverse the stem cell phenotype. So the other compound I want to talk about is this GSK3 inhibitor. There's the inhibition curves with the cohesin mutants and wild type. And where this actually hits the Wnt signaling pathway is this complex here that normally targets beta-catenin, the effector for Wnt signaling for destruction. So when this complex is operating normally and you have no Wnt ligand, beta-catenin is targeted for the proteasome. If you inhibit GSK3 beta-catenin, this complex doesn't work anymore, and beta-catenin is around and gets to go to the nucleus and switch on Wnt genes. So, so this actually agonizes this whole pathway. So we did some experiments. We took our leukemia cells with STAG2 mutations and stimulated them with Wnt to see what would happen. And what we found is that we get many more genes that respond to Wnt than you do if you don't have the mutation. So you can see here, nearly twice as many genes are upregulated when you stimulate these cells with Wnt than normal. And then if you look at kind of what sort of genes they are, some completely new genes that didn't even care about Wnt previously suddenly become activated when you have a STAG2 mutation. So yes, so these cells are definitely precociously responsive to Wnt. And then we wondered why. So we looked to see what was happening with beta-catenin, and it turns out that it tends to be stabilized in these STAG2 mutant cells. So this is a fluorescence. This is immunohistochemistry here that shows that in SMZ3, STAG2, and RAD21 cell lines, you have these punctate stabilized beta-catenin complexes. So the cells are already kind of primed to respond much better to Wnt agonism. And that's just the quantitation of that. So if you apply the GSK3 inhibitor, you can then see that the chromatin, according to this histone mark H3K27 acetyl, becomes a whole lot more open. So here is the normal cell line. Here's our favorite, RUNX1, right? Stimulate with this is a different Wnt agonist, sure, and you get more open chromatin, including at RUNX1. But if you have a STAG2 mutant, this becomes a lot more pronounced and the chromatin is more open, and so the gene will be more responsive. And back to our favorite zebrafish, we have Wnt reporter lines in zebrafish that glow when you give them CHUR or bio or something that agonizes Wnt. And here we're focusing on some cells in the midbrain. Here's a wild type fish, and then my lovely NZ171 or the 17.1 mutant. Cells in the midbrain, many more of them become more glowy when you stimulate them with Wnt. So meaning that the cells are much more primed to respond to Wnt, and cells that wouldn't normally respond. So more recently, and this is, I can't really resist a little bit of developmental biology. So this is actually now taking another part of the zebrafish tail bud, which is a population of cells that are differentiating constantly into neuronal or mesoderm cells. And we're asking questions like, so what does compromising cohesion do to cell fate decisions in the tail bud? And what we find is so this is single cell RNA-seq now, a UMAP of tail bud cells. And this is a UMAP showing which cells, which cell types are much more different than wild type. And it turns out it's mesoderm cells that are more different than wild type. And when we looked at these cells, well, what's different about you cells? Why are you different? We found that they had different Wnt gene expression in them. So these are the neuromesodermal progenitors. There is actually down regulation of Wnt signaling in these neuromesodermal progenitors, as shown in this kind of box here. So particularly Wnt11 and Wnt8 is changed. So that means the directionality of Wnt signaling is a little bit confusing to us, but we can certainly show using animal models and cell lines and all these different kind of parameters that Wnt signaling is somehow quite dramatically changed when you have a cohesin mutation. And if you use a Wnt agonist, somehow that causes these cells to crash and die. So why is that? So let's go back to what cohesin does in the cell cycle: gene expression and cell division. So BRD inhibitors, they affect chromatin, they dampen active enhancers. So I think we can be reasonably confident that our BRD inhibitors that we found in the screen are operating by dampening the chromatin defects that are in cohesin mutants. So where we have active histone marks, those will be swamped down by BRD inhibitors, and so you will reverse those effects. But what about Wnt agonists? You might well think, well, maybe that's probably transcriptional too, right? Because Wnt signaling beta-catenin switching on genes. And so, yeah, that's what I thought was happening for a while. And then it's always good to read papers, isn't it? Because then you get a lot of different information. So I started to read things that I wouldn't normally read, and it turns out that GSK3 inhibition leads to mitotic defects. And so there's not huge numbers of papers, but there's like a half a dozen papers that show you that if you inhibit GSK3, you'll get mitotic catastrophe. So what I now think is going on with the GSK3 inhibitors is that we're actually exacerbating cohesin cell cycle phenotype. So deeper reading of these papers showed that what GSK3 inhibition does is make S phase happen faster. and that induces replication stress. So you've got a lot of replication going on, you can't keep up, and so you possibly get mitotic catastrophe. You especially might get mitotic catastrophe if you don't have enough cohesin complexes or you've compromised cohesin. And then you'll get this sort of stuff going on. So what we think is probably going on with the Wnt agonism is accelerating replication such that you get replication stress and then you cannot deal with it, crash the cells and they die. So, in summary, so the implications for leukemia, there are deeper implications for Wnt signaling in leukemia, and that you need Wnt signaling to maintain hematopoietic stem cells in the first place. So you need it for self-renewal. When you reduce, we haven't shown this, but other labs particularly in the US have shown that when you reduce cohesin function, you get HSC expansion and a block in differentiation. So it could be in part due to enhanced Wnt signaling that you get this phenotype with a cohesin mutation. But when you get the sensitivity to pushing the Wnt further, it's probably because of the mitotic consequences of GSK3 inhibition that you get this. And so in general, what our lab has shown over time is that you don't get this hardwired enhancer promoter looping owing to cohesin having this role, and you get dramatically altered chromatin at baseline in cohesin mutant cells. If you get a deficiency, it's more like you deconstrain transcription. So you can gain gene expression, you can lose gene expression, and the high schools that had the fence between them are now able to mingle with each other, and all kinds of stuff can happen. So some of the spurious transcription you can reduce with BRD4 inhibition. I wonder if the cell cycle and gene expression roles can really truly be separated, because some of the inhibitors that we thought might affect transcription actually hit the cell cycle instead. So in summary, I'd like to really acknowledge a lot of the people who did the work. So Jetta Anthony in my lab has been a stalwart in the lab for the past 11 years, and she's done pretty much anchored all the cancer cell work that you saw. A lot of it's not published yet, so I'm trying to get her to write that up. The synthetic lethal screen was Alice Chin's PhD, and then some of the zebrafish stuff was performed by Anastasia and Sharada, who was previously in the lab. And I'd like to acknowledge too the collaborators in Australia and some also collaborators at Otego that helped advise on the synthetic lethal screen. Of the funders, I'd particularly like to acknowledge the Fulbright Foundation of New Zealand. So I was here on sabbatical in 2019. It was meant to go some distance into 2020, and you can imagine what happened in 2020. I had to go home. So I'm here because I had some Fulbright money left, and so I was able to travel here to speak to you today. So I'm very grateful to them for the ability to share these results with you. And on that note, I'm happy to take any questions if anyone has any.
So, forgive me, this is not my forte, but you may have already explained it. What direction do you see your lab going with forward? What are you going to do with your data for future investigations?
That's a really excellent question. So I think we found out all we can in the cell line models, and where things are going in the community is really patient-centered genomics. And large collections of wikipedias with deep sequencing and phenotyping of those wikipedias. This is not something that we can do where I am in New Zealand. I mean, we're much more suited to kind of answering niche questions, if you like. And but we do have a new project in the zebrafish, and this is to look at some of the properties that cohesin cells gain, deficient cells gain when they have cohesin mutations. And I mentioned just briefly, what happens in the cell cultures is that the cells start to get sticky and they sit down on plastic and they gain stem cell-like properties. And so now we're wondering, is do they gain leukemia stem cell-like phenotypes that help these cells engraft in the hematopoietic niche? And does that protect cohesin deficient cells from normal therapies? So we're testing this in a zebrafish model where you can actually take out a kidney. Well, so pretend the zebrafish kidney is a bone marrow, because it is, that's where the blood cells are in the zebrafish. You can make a suspension, you transplant that into another zebrafish, and those cells will engraft in the zebrafish kidney. And because fish are cool and you can label them with fluorescent colors, you can make those cells green and fluorescent and watch them engraft in a host kidney. So you could take cohesin deficient cells that are green and wild type cells that are red and compete them against each other to engraft in a zebrafish kidney. And then you could try out your different drugs and see which ones win against which cell types. So that's where that work is going next. Another work, we're using single cell approaches to look at cell fate decisions using other parts of the zebrafish and trying to understand cell commitment principles of cohesin. Yeah, so that's really where things are at and going, I guess.
I have two questions. Just curious about the history of the protein RAD21. Given the name, I'm assuming it was picked up in some kind of radiation screen.
Absolutely.
Have you seen later on if it's associated with the double-strand breaking point or something like that?
It is.
And then the second question is, were you surprised that in your screen, aside from aurora kinase, there weren't any more inhibitors associated with cell cycle checkpoint inhibitors or mitotic, things involved in mitosis? Because so many of them appear to be more classical kinase associated pathways. So I'm just curious if you were surprised by that.
Yeah, so I could have mentioned to you that people have done similar screens and identified PARP inhibition as being synthetic lethal in cohesin mutants, and that is double-strand break inhibitor. Yeah, so on your first question, I oversimplified to say that cohesin had cell cycle and gene expression roles. It has a third very important role in damage repair. And this can happen during the cell cycle to repair the breaks that happen, in G2 and repair that. You need cohesin complexes for that, but it can also repair in interphase as well. And you're right, RAD21 was identified in yeast in a radiation screen as a gene important for DNA repair. So it has that role and it involves ATM and that whole pathway. And then PARP inhibitors, we tried as a sort of a positive control in our screen. And right down the bottom of my gene list, there was a PARP inhibitor there. It did come in there, but it wasn't in our top ones. And I don't know why. And it's possibly because we have hits and not wild type cells for the RAD21 SMC3, and it's possible that STAG2 itself is not needed for double-strand break repair. You just need the core complex for that. So, yeah, hope that answers your question.
Last question. Yep. First off, great talk. I really liked your description of all these techniques to get to the mechanism. My question has to do beyond the germline somatic mutations. A lot of your drug candidates, you used to have GSK, but you also had AKT and mTOR on there.
Yes.
Is this mechanism also present in cancer susceptibility in conjunction with diabetes and, say, obesity?
That's
as far as things
I don't know. That's a really great question. I don't know the answer to that, but I wonder. One of the aspects I'd really love to look at, if I could kind of start a whole new thing, would be to look at cellular metabolism. And how that tracks with cohesin mutation, and because you got MEC there as well, and you've and all of this sort of things linked with growth there. So that's an excellent question, and I don't have any good answers for you, but I think that because these things turned up on the screen is actually pointing us in a direction that this is all very much linked with metabolism. Which again is why I wonder if you can really tear apart these things to do with cell growth and division and gene expression. Thanks for the question.
Maria, I want to thank Dr. Horsfield.
Yeah.
Thank you very much.
Sorry, I'm
Sure.